Good afternoon and welcome to our second segment of Nutrigenomics, the bridge between nutrition and genetics. This is a portion of our continuing education program through Antioch University. Together over the course of the past few weeks, we have been learning about nutrigenomics and its impacts. Throughout these lessons, we have recognized the multitude of factors that play a role in shaping gene expression. Some of these roles have been environmental-based, some of them have been individual lifestyle choices, while others are overarching impacts such as social, economic, and political factors. Overall, mental health professionals have significantly underestimated the importance of lifestyle factors as one contributors to and treatments for multiple psychopathologies. In addition, these lifestyle factors are also important in fostering individual and social well-being. And lastly, these factors are important in preserving and optimizing cognitive function. Consequently, therapeutic lifestyle changes are underutilized despite considerable evidence of their effectiveness in both clinical and normal populations. Therapeutic lifestyle changes are sometimes as effective as either psychotherapy or pharmacotherapy, and can offer significant therapeutic advantages. Important therapeutic lifestyle changes include things like exercise, nutrition, diet, time spent in nature, interpersonal relationships, recreational activities, and our time spent on relaxation and stress management. So, of course, stress is inevitable. Many of our lives have countless stressful attributes. However, our abilities to de-stress, to relax, and unwind after stressful days are quite important. And the last one to consider too are community services. So sometimes these give us a sense of purpose and connection to the community. Could be something such as volunteering at a community garden or participating at a local food bank. And these factors are important to balance with other lifestyle attributes, but can often be integral, especially with folks that are retired and still looking for having that connection to the community and sense of purpose. Moreover, research is investigating their effects and effectiveness, the principles, advantages, and challenges involved in implementing them. Some of the attributes to consider are economic, institutional, and professional forces at play that may impact accessibility. Where possible, therapeutic recommendations should be communicated in an easy manner to understand. This is primarily because clear communication strongly influences whether therapists recommend practices and patients adopt those interventions. So communication is key. And if that isn't easy for therapists to communicate, to give those recommendations, then it may be difficult or add additional parameters that prevent patients from adopting and implementing those interventions. We are all unique in our own ways. There lies countless wisdom in this statement. In eliciting this school of thought, advances in molecular and recombinant DNA technology have led to exquisite studies in the field of genetics and the recognition of a much more specific way through DNA sequencing and how unique each and every one of us is. The extent through which genetic variation occurs really lends some insight into the elegance of the body systems. You could take a look at any individuals, no matter how closely or distant related they are, and find incredible genetic variation. In science, we sometimes call these factors inter-individual differences, and they play important roles when providing recommendations, and in this case, through DNA sequencing to truly understand how unique each and every one of us is. Considering that a personalized diet based on nutrigenomics could benefit public health because of its ability to motivate different consumers to change their dietary habits. What are some ways you can think of through which we may unite multiple disciplines together to tackle chronic disease prevention across nations? When we think about chronic disease prevention, we really have to take into consideration the inter-individual differences, the uniqueness among every single person. Remember, when we first spoke about genetics, we touched upon the fact that we have about 20,000 genes. It's a whole lot of differences embedded within those genes that account for the different characteristics, the different prevalence, and the predispositions that each person may or may not have or express. But when thinking about chronic disease prevention, we really want to try to tailor some of our recommendations and approaches to account for these inter-individual variations or uniqueness that each and every one of us carry. Moreover, considering nutrient-gene interactions, there are three major conceptual groupings for thinking about. So when we consider the way that the food we eat or our nutrients interact with us at the genetic level, at the cellular level, there are three schools of thought or forms of interactions that come to mind. The first are direct interactions. This is how the nutrients that we consume through food, through our dietary habits, can interact with a receptor. Now, hearing the word receptor may be the first time for you, so I would like to provide an analogy. Consider the lock and key of a door. So the key that you have has been carved in a particular way so that it unlocks that door, and only that key will work for that specific door. Similarly, we have receptors throughout the body that are specialized or shaped to only accept certain nutrients. So certain vitamins or minerals can bind to those receptors, just like a lock and key of a door. Well, when they bind or interact, they behave as transcription factors and can bind to DNA and induce gene expression. The second mechanism is through epigenetic interactions. This entails nutrients being able to alter the structure of the DNA. So the actual structure or the makeup, the composition of the DNA, or of histone proteins within the chromatin. So that gene expression is chronically altered, so altered across the long term. And then the third interaction is through genetic variations. Common genetic variations can alter the expression or functionality of genes. When you hear the word genetic variation, you may have also come across the term single nucleotide polymorphisms. These are often shorthanded as SNPs, SNP''S. Well, these single nucleotide polymorphisms, consider them as one alteration or one morph, one change. So for example, if you had a recipe book and you were following that recipe book to create your favorite meal, but you swapped out one ingredient, you changed one thing, a single morph. Or for my coders out there, if you took a computer coding system, and you changed one code, that would be similar to a single nucleotide polymorphism. It might give you a different output if it's a computer system, or if it's a recipe, it might give you a slight change to the taste of the meal by changing one ingredient. Well, similarly, when we see these genetic variations, they can alter the expression, or in other words, the functionality of the genes. If you think back to our foundational knowledge of genetics, there are 4 nucleotide bases that are used: A for adenine, T for thymine, C cytosine, and G guanine. If you change out one of those nucleotides, for example, swapping a G for a C, a guanine for a cytosine, that could be a similar way to think of a single nucleotide change. All of these mechanisms can result in altered metabolism of and altered dietary requirements for nutrients. In the past 2 decades, physicians, geneticists, and nutritionists have begun to study the effects of genetic variation and gene-nutrient interactions in the management of chronic diseases. Some of the leading chronic diseases that you may be familiar with are coronary heart disease, hypertension or high blood pressure, many forms of cancer, diabetes, type 1, type 2 being the one we're referring to in terms of preventable chronic disease, and obesity, which we have seen increase in prevalence across the years. The role of nutrients and the impact of gene expression have an influence upon our chronic diseases and the rates of prevalence. a new era is being ushered, and in which is called nutrigenetics or nutrigenomics. This new field of genetics has enormous implications for nutritional research, both in the prevention and management of chronic diseases. So consider if we could take a look at a person's genetic makeup and elicit their predispositions for particular diseases, such as coronary heart disease. And based on that information, what if their medical professionals could provide them with tailored therapy or tailored mechanisms to prevent the onset of that chronic disease? The side of prevention has been shown to be far more successful than treating the patient once the chronic disease has already taken root and taken hold. To add on, to layer on, families or people that are related share both genes and environment, and similarity may result from either. Much research has been carried out to define the contribution of each and their interaction in the development of the individual. So considering hereditary aspects are also important in this field of work. Knowing a person's susceptibility to a particular disease helps to determine their risk factor. More so, genetics help us predict their response to a particular diet. Through dietary treatment, the patient may gain the most therapeutic benefit. Currently speaking, the costs of genotyping as a whole have dropped markedly. However, the number of markers available per gene makes the genotyping of all markers still expensive. So in terms of future implications, if we can decrease the cost of genotyping as a whole, we can make this field of work more accessible for people of all socioeconomic factors. While there are countless benefits to pharmaceuticals, perhaps this newer field of nutrigenomics may elicit other options for patients to consider. Future implications demonstrate the responsibility of the nutrition scientists to bridge genetics and nutrition to establish nutrigenetics as a major discipline in our leading fields of science. I appreciate your time today. Research that was shared came from the following works cited in references, and I welcome any questions that you have. Thank you.